So today I'm going to share with you some of the research that my lab is doing that's focused on understanding who is the beneficiaries of genetic research studies and how does that translate to who gets benefits in the clinic. And so what we are trying to do right now is figure out what can we do to address those disparities even without collecting additional data. And so we are a human genetics lab. We focus on studying human genetic variation. I'm often quite jealous of my colleagues that have a lot more control over their systems. They get to control what their mice eat. They can mutate their genomes. They can see what happens. I always tell everyone I can't do any of that. I can't control any of the people in my study. All I can do is take advantage of all the things that have already happened. What we're doing as a human geneticist is trying to leverage what's already existing in the human population and then pull insights. And so you can imagine how quickly confounders can come into play here. The very standard way that human geneticists try to address this type of confounder, not knowing who slept well last night, what you had for breakfast this morning, whether you made a good choice or a bad choice in terms of exercise. Without being able to control for that, what we do is just collect massive amounts of people, trying to diffuse out those variables so that we can determine what are the genetic connections to traits despite all of those confounding variables. And so this requires pretty large sample sizes and, as you can imagine, very expensive research studies. In total, though, we're looking at a very small fraction of your genome. You're about 99.9% similar to every person in this room. Only 0.1% of your genome is going to be different. But the genome is quite large, so that represents about 6 million base pairs of difference. So when we're trying to figure out what are the genetic changes that make someone develop schizophrenia versus depression, we have to look amongst these 6 million base pairs of difference and try to pull salient conclusions. The way this is done typically is through these large-scale association studies. These studies are quite simple on the surface. We're collecting large groups of people that have a trait and large group of people that don't. We then collect all of their genetic information and assess genetic variants one at a time. Do we see an association with a particular genetic change in people who have the disease versus those that don't? So we're going to look for genetic differences that are found at increased prevalence in the disease population relative to the controls. These are then said to be associated with disease. Now what's tricky here is this may be different from some of the genetic information you've come across before. Maybe you've heard about Down syndrome, which is caused by an extra copy of a chromosome. Every person that has an extra copy of that chromosome has Down syndrome. We haven't found a single human where that's not true. But for the majority of diseases in the human population, that is not a simple pattern. So there's typically many people that have different variants that cause the trait we're looking for. So what we're thinking about more is not a genetic cause of a trait, but a genetic change that increases your risk or likelihood of developing disease, just like smoking increases your risk of lung cancer. And so these large-scale studies go through and identify DNA changes that associate with increased risk. And then the goal of the biologist is to understand why those variants would cause you to be at increased risk. Could that help us figure out how to prevent the disease, treat the disease, or even just diagnose it so we know what we're looking to affect? So this type of study has been performed for about the last 15 years. Here's what we've gotten so far. Each one of these bars represents one of your chromosomes, and the little dots are representing DNA variants that have been linked to a trait. These are all of the results we had up until 2008. By 2011, we had more, and then 2015, 2019, by 2020, we're getting off the scale of my plot, and I had to downsize to get enough pixels on this screen so that we can see all the results we have up into 2024. This is what happens when sequencing studies start exploding, and people become more interested in understanding their own genomes and consenting themselves into genetic studies. What this represents is 600,000 clues about how diseases work, if only we could interpret them. So this is a huge amount of information that many groups, including my own, are trying to figure out why would this set of variants increase your likelihood of developing one disease versus another. What's the problem? The discovery cohorts are incredibly biased. The vast majority of that data that I just showed you comes from the European ancestry populations only. So about 80% of all of our genetic studies are performed only on European ancestry. And this has led to pretty big disparities in the clinic. For example, we have some genetic variants that we can use to predict who's likely to develop colon cancer that are incredibly effective in Europeans and completely ineffective in those of Asian descent. So we're thinking about who's benefiting from all this genetic research and what can we do to translate that information is a major problem for the human genetics field. And this is a well-recognized problem, as the NIH has put substantial efforts into increasing genetic diversity. We're now seeing massive studies being performed across different continents across the world, and more of a concerted effort to engage with populations that may have been reticent to be involved in genetic studies. But this takes time and money. Collecting thousands and thousands of genomes takes decades, takes a very long time, and allows you to finally fully pull some associations. In the meantime, what kind of things can we do? Can we start to think about different ways that we analyze the data in order to pull information from some of these smaller studies that represent these populations that are pretty much missing from the benefits of genetic research? Not only are some populations missing, some diseases are also underrepresented. So you can imagine where voting in Congress and their choices about NIH funding has also influenced all of these results. Breast cancer, very well studied. Alzheimer's disease, very well studied. Opioid addiction, not so much. Right? So there are some traits that have historically not been as well funded in the federal research, which has led to these cohorts being underrepresented. And so our goal here is to try to develop methods that allow us to pull insights now instead of waiting for new data. And the way that we go about this is trying to think more deeply about how the genome functions in changing the models to find effects despite not having huge power. So one of the areas that we focus on specifically is opioid addiction. This is a pretty critical public health crisis. Since 2017, opioid overdoses have been the leading cause of accidental deaths, even surpassing that of automobile accidents. And this was only exacerbated during the COVID-19 pandemic. So it's a massive problem, but you may be asking yourselves, is this a genetics problem? Right? So is addiction a genetic trait? And we can ask that question by looking at twin studies. So take advantage of a very naturally occurring phenomenon where the same genome is born into two individuals, and ask, do you see a correlation in their phenotypes? Do you see individuals who were born with the same genome but raised in separate environments having the same trait? And in doing this type of analysis, you find that opioid use disorder is about 60% heritable. Height is about 90% heritable. Schizophrenia about 80%. So it's right along these lines, maybe a slightly larger environmental component, but still clearly a major role for genetics. But the problem is that we have not discovered what are the underlying genetic causes or contributors to opioid addiction. For most diseases, we're talking about hundreds of genetic risk factors have already been identified. For opioid use, we're maybe 5 known risk factors, and they're all involving the actual opioid receptors in cellular processes. This has largely been attributed to sample size and power, but also some of this is due to the fact that addiction might just be a more complicated trait. More variables are at play that make each individual with opioid use disorder slightly different. Different causes would make it very difficult to pull together, especially in small cohort studies. So you can think of opioid use disorder and other related types of substance use disorders as multiple stages, where first there is an experience of exposing to the drug, a binging period, typically followed by withdrawal symptoms leading to relapse and recreating this cycle over and over again. In each stage of this disease, there are different opportunities for genetic risk factors to come into play and different causes to cause the trait. So for example, who's going to be exposed to an opioid to begin with? Well, there are genetic connections here to someone's impulsivity or willingness to take risks. There's actually genetic factors underlying that. Ease of access, so there's some social factors. Some people are not going to be prescribed an opioid in the clinic, and some will be more likely to. Some may have higher pain tolerance and say, after that orthopedic surgery, I do not want to take the opioid. I'm going to stick with ibuprofen. Right? So there may be differences at all those levels that have different genetic correlations. We also see massive variation in one's response to opioids. Maybe you've been prescribed an opioid before. Some people experience euphoria. Some have pretty nasty side effects and don't want to continue taking their dosage. So it really depends. And it turns out if you enjoy your first opioid experience, you're 20 times more likely to have addiction in the future. So if you have anyone in your life that has ever been prescribed an opioid and describes a very happy feeling afterwards, maybe help them monitor their intake until their prescription has run its course. The other challenge here is the withdrawal. Some people experience much more severe withdrawals, making them much more prone to relapse. And that's incredibly problematic because that is where overdoses tend to occur. So it's in this relapse phase that we see the majority of opioid overdose deaths occurring, rather than like the first exposure stage. So when you're talking about a trait that's not only understudied, but multifactorial with distinct causes, you can see where the challenge lies and why we haven't made much progress in understanding the genetic risk for this trait. And so we've been tackling this question looking at these smaller disease studies, but trying to account for this heterogeneity, this mixture of causality. And so we can study genetics by looking directly at the sequence, but we can also study genetics by looking at the output of DNA sequence. We can look at the genes and what kind of products they make, the RNA molecules that are produced, the proteins that are produced, and then a specific type of element that my lab studies, which is the element that controls that process. So your body has to dictate which genes are turned on and off in different contexts. It's one of my favorite things to teach the undergraduates because it's something they know, but they don't know that they know it, right? You only have one genome. You have basically the same genome in every cell of your body, yet your heart beats, your neurons send impulses, your immune cells fight pathogens, all coming from one single blueprint. And so understanding how that happens is all about controlling which genes are turned on and off at what time. And so this process called gene regulation is what we focus on. It's this effect of these little triangle represented elements that decide when those RNA molecules are produced. Turns out majority of our genetic risk is actually coming from disrupting these regulatory elements and not the proteins themselves. And so our approach was to go directly to patient tissues and capture these gene regulatory elements and ask how they are disrupted in addiction. We collected about 100 individuals who either died of an opiate overdose or died of an accidental death, typically a motorcycle accident, to compare their brain tissue samples, and did an experiment that allows us to look at those regulatory element activity. To give you a sense of what the data looks like, this is one tiny fraction of the genome of your 6 billion base pairs. This represents about 20,000 base pairs right here. And these little squiggles represent which gene regulatory elements are active. How you interpret this data requires a number of different strategies. So we're not going to go into too much detail, but I always think it's interesting to see what these traces look like. And how do you go through and look at the entire genome and decide what differences are actually interesting or valuable to study? The standard approach is to try to go through and identify something that is consistent, something that is different in everyone who has the trait, that the controls all have something different. So for example, we'd look for an element where the big triangle peak here is high in all of our controls and flatlined in all of our cases. You can use a lot of straightforward modeling, some linear regression type analyses to identify these consistent changes. This often results in very little information from small studies because you're requiring consistency. What we wanted to do is allow for that heterogeneity. And so instead of performing these sort of standard strong impact analyses, we look for changes that are specific to each individual with opioid addiction. So say, of our control population, this is what we expect to happen. Is there any novel information, any surprising information in the one sample that has opioid use? So we're performing this very highly individualized analysis. So here's a little cartoon view of what this looks like. This is what standard approaches look for, a small reduction that's consistent in hill height or triangle height in this cartoon. What our approach does is looks for changes that are more akin to here, where it's consistent in the controls, and then one individual has a big change that's really unexpected. We can go through and define these types of changes for every individual and look at what comes out of it. And as you might expect, when you apply an analysis that looks for individuality, most of the things are individual specific. Only about 10% of these changes were found in more than one person with opioid addiction. But what the way is interesting about how these elements work is they don't work independently. So these individual elements that are controlling genes, they actually work in combinations. So all of these little triangles here are responsible for controlling the same gene. And so we start by linking all of these differences back to the genes that they control, and then we suddenly see a lot higher degree of convergence across our opioid cases. So of the elements that we identified as being individual specific, only 10% were found in multiple patients. But if we focus on the genes that are affected by those little triangle changes, then we see that happening in 60%, happening in multiple patients. So what we've been working on with the help of the SEED grant is how to formalize a convergence analysis. How do we quantify the areas of the genome where there's the most overlap or grouping of these individual specific changes? We started applying this to our opioid cohort and found 5 major genes linked in our first brain study and 50 additional genes in our second brain study. We're really excited about these results. We started looking at these in cell culture, so in neurons growing on plastic. We're seeing some very interesting insights about the role of insulin signaling in addiction. So with this analysis, we're now formalizing to enable us to think about these target genes and apply them to other traits such as schizophrenia and autism. So that I'll wrap up. So this is a work across a number of individuals, and I want to make sure I highlight the cohort of individuals that allows us to collect this data. We do not do any autopsies or dissections. We have collaborators that collect the data so we can focus on the computational analysis, and that's this group of researchers here. So thank you very much.
Thank you so much. Questions for Olivia?
Thank you. This was fascinating and very well presented. My question is, I applaud you for looking at opioid addiction. It's such a huge problem, but it's also a highly stigmatized one. Yeah. And my question is about the ethics of, like, once you have actionable data that could be useful in a clinical setting, how would you work with the ethics around that? And particularly in this moment where we have a lot of mistrust of science and things like the 23andMe acquisition are really making people very anxious about what people might do with genetic information about them. How would you think about that, and where would we even begin?
Yeah, it's a great question. It's something we think about a lot because of all the diseases we study, this is the only one that's fully preventable, right? So never exposed to a substance, you're not going to develop addiction, right? But there's a huge risk of applying this type of result to the clinic without thinking about those types of boundaries. So say we could predict who's at highest risk for addiction. Does that mean they shouldn't be prescribed a painkiller? Probably not, right? Until we have some new alternatives. That's some very exciting new painkillers are on the market now, so hopefully we can start phasing out prescription use. But what we want to think about is ways of increasing monitoring. Maybe if you're at high risk, it's not that you don't get access, but you'll get a week at a time prescription instead of a month at a time prescription, so more intervention. Some places, I think it's common in Texas, for example, to not prescribe opioids in the orthopedic surgery center, but to refer them to a pain clinic. So that's just this extra step of having to contact a pain clinic to talk to a doctor to get access to an opioid actually reduces overall opioid intake. So there are ways in which we can monitor this, sort of making sure that people have the knowledge, but also the policies to keep how we would restrict access very carefully selected. We also have to be super conscientious of the way we design these studies. The very first machine learning model for addiction predicted race and not addiction. So you can imagine how quickly that could be problematic if applied in the clinic.
Great.
Hi, I had a question if you were considering also histonic activity or transcription factors, because you said regulatory elements, but I'm not sure which one you implied with that. And also, if you're talking about one or the other, are you considering the activity of the other one?
Yes. Yeah, so we're actually measuring histone modification. So when I talk about a regulatory element, we're measuring the proteins that are altered that control how the genome is folded. And what I didn't have time to go in today is we started predicting what might be driving these changes. And so we have a few transcription factors that we think are candidates for causing these effects, and then they might be better therapeutic targets because they cause many downstream effects. So good question. Yeah.
Any further? Yes. And then afterwards, yeah.
Thank you. I understand this research is heavy on genetics, but I was wondering if this research accounted for environmental factors at all?
Yeah, so a lot of our models attempt to, right? We look at things like age, the substance that you're using. That's one of the hardest things to control for. Most people with opioid addiction are polysubstance users, so they're using fentanyl as well as heroin or some mix thereof. And so that can really be a major confounder. Age is another big one. Surprisingly, the biggest confounder is something called the post-mortem interval, so the time between death and tissue collection. And so we can try to model that effect out, but we have to be very aware that that might be a driver.
Hi, absolutely love your presentation. I just had a question about the causes of these addictions. As we know, with a lot of addictions, that childhood trauma and childhood abuse and various factors lead to addiction. And I was just curious to know if you factored that in your computational analysis.
Right, so the benefit of looking at human tissues is that you capture everything that occurred in a lifetime. So some of the things that we look at may be a direct effect of opioid exposure, and that's why the brain DNA organization has changed. But they could also be pre-existing opioid exposure. So we're looking, currently we're starting exposing cells on plastic to opioids and seeing what changes in that very simplified system. And what it looks like is in one of our brain cohorts that we've studied in the frontal cortex, that is where you see more signs of these effects being early childhood adversity related, so high stress in early childhood can change the way in which the brain is organized, the epigenetics of the brain, and has pretty lasting impacts. And we see some evidence of that here. So the postdoc who just started that's looking at how the effects of stress in early development may influence response to opioids.
Awesome. Further questions? Yes.
Hi, thank you so much for your presentation. My question was going to be about data being biased. Is the data gender biased? I think the data that is coming from the past is most of the time male dominated. Is it something that we should be cautious about?
Yes, so we have, most of the studies that have been published have been cognizant of gender differences, of sex differences, because we're collecting X and Y chromosome related information. We can see that very clearly. It's in the follow-up that you see the most disparity. So which loci you're studying mostly are studied in mice, right? It's when we follow up on an experiment, and that's mostly done in male mice. But the actual discovery is pretty well balanced with some notable challenges. So one of those strange challenges for us is that it's hard to find accidental death female controls. Women tend to die of chronic diseases and less likely to die at a young age of an accidental death like a motorcycle accident. And so it's a great thing that we can't find enough controls of accidental death, but it's also a challenge. So we have to be very aware of that so that we're not misrepresenting women with addiction in the study.
Great. And final question.
Yeah, I'm a statistician, so forgive me if this is too naive a question, but you said something very interesting about complex traits, namely that I'm assuming complex traits here are the more the number of base pair differences or something, or combinations thereof. And interestingly, those are also precisely the traits which perhaps occur in fewer people. Like the more complex the trait, I'm assuming there are fewer people in the population that have that trait.
It's inverse, actually.
Oh, I see.
So the more, if there is a trait that's caused by a single major effect, it's more rare because that effect may be selected against with evolutionary pressures. But when it's complicated, you have many different little causes, they can live in our genomes for longer. So it's actually the most common traits that are the most polygenic, or have the most genetic factors that influence them.
Oh, okay. That's good to know.
Yeah.
Okay, thanks.